In the biopharma industry, successful drug discovery and development is crucial. This success is dependent on identifying relevant genes targets for the diseases of interest for the company, and then ensuring that drug discovery efforts are focused on a few specific genes and high impact targets. In order to support these efforts by using a data driven model, I wanted to come up with a list of genes that could drive drug discovery efforts. My approach is focused on homologous mouse genes that are associated with breast cancer. Incorporating some additional feedback, I narrowed this further by adding an additional data set with gene expression data that had quantitative data for patient treatment methods. This also allowed me to expand the outcomes of my data analysis to include not just these reports of gene associated with diseases, but more specific information about gene expression levels in breast cancer and correlation of those genes with survival data, treatment methods, and age of diagnosis. I wanted to use a clearly annotated data source to narrow down my business need to one disease state and a concise list of genes. So the data source that I decided to utilize were the gene, disease, and phenotype datasets from Mouse Genome Informatics database. These datasets comprise of more than 100,000 genes that are annotated to disease and phenotype ontologies. I could then pull a list of more than 2,000 genes that had specific impact on human diseases, from which I was able to conduct my initial analysis and come up with a narrowed down list of 10 genes with varying levels of relative expression in breast cancer. In order to further correlate the final list of genes associated with breast cancer with outcomes, I also utilized the MetaBreak dataset from Kaggle.com, which gave me gene expression data for breast cancer genes along with patient-specific information, including age, treatment methods, and overall survival. I used several statistical methods within this analysis to come up with some conclusions about data. I used cumulative probability to predict the importance of gene expression on the effectiveness of therapy as well as gene impact on survival outcomes. I also used the mean and standard deviation of overall survival in months to predict the likelihood of a positive response to chemotherapy, radiotherapy, or hormone therapy. I utilized the hypothesis testing method by changing the variable type from numerical to factor for the different types of treatments, then looked at the relationship between expression level of genes of interest and the efficacy of those different therapies. Finally, I utilized linear regression to evaluate the relationship between overall survival in months for different therapies and age diagnosis and expression levels of genes of interest. There were alternative approaches that I considered while designing this project that ultimately were rejected based on the data that I was working with. I couldn't utilize regular probability as the output was individualized for each of the 1900 patients, which would not have been helpful for making a rational conclusion. I also had to reject the use of cluster analysis for gene expression data as all the data points were clustered together when I factored in additional parameters like overall survival and age at diagnosis. I did not have sufficient parameters to be able to conduct logistic regression analyses. Therefore, this approach would have also failed to provide any actionable outcomes related to therapy efficacy or pattern in overall survival in rational gene expression. The importance of this solution lies in the narrowing down my data set to a list of 10 genes that have been determined to be associated with better outcomes in breast cancer in the presence of certain therapies. In order to accomplish this, I followed the steps that were initially outlined in our business intelligence roadmap. I ended up adding a few additional steps and data sets to the initial layout. Using this process, I was able to come up with a list of genes and justify their usage by using a statistical analysis. This list can be used now in our drug discovery efforts by focusing on therapies that are targeted to these genes. We can also utilize this information to identify patients that have high expression of specific genes, potentially selecting for those that may benefit from specific types of therapies. This methodology allows us For a concise list of genes to be collected from a dataset, they started with 100,000 genes, greatly streamlining efforts and optimizing the drug discovery process.